I'm going to talk today about our Texas horned lizard reintroduction project. And this has been a long-term project for us with many partners. So I'm kind of going to give an overview first of horned lizards in general, and then kind of run into what we've been doing the past 15 or so years, and then how we manage the horned lizards in captivity, as well as kind of where we're at now, what our current projects and focus is. So I kind of want to start out first by introducing the character on the left-hand side of your screen. I think a lot of you probably already know or have seen this mascot from Texas Christian University, TCU. That is one of our major partners. He comes down and he has visited the zoo. He's holding one of our little horned lizards in his hand, and I wanted to dispel some myths. First off, he goes by, I think Super Frog is the original term or name of this critter, and I think he's now the Hypnotoad. I'm not exactly sure where that came from or why that is, but that's what he goes by now. But the horned lizard is indeed a reptile, so it's a lizard. It has scales, it has movable eyelids, and it has all kinds of cool features that enable it to survive in very dry, arid habitats. And of course, toads and frogs are amphibians. They have very semi-permeable skin. They rely upon moisture and being around water environments. So just want to make sure that everybody knows the difference between the reptiles and the amphibians. And, you know, folks in Texas have been calling these critters horny toads for over 100 years. So that is never going to change. It's kind of a term of endearment, and I realize that. But just moving forward, I want everybody to know for sure that they are reptiles, because sometimes people do come here and they're a little confused thinking they're frogs. So anyway, so horned lizards in Texas, we do actually have 3 species of horned lizards in the state. They have the round-tailed horned lizard, which is the smaller one, actually really cool, excellent at camouflage. They come in a lot of different colors within one clutch, but they possess 4 medium-sized horns that are lined on the back of the head, and this is kind of how we determine which species of horned lizards we're looking at, is by looking at their heads and their horn structures. And they come from the rocky areas in the western part of Texas. And there's the greater short-tailed horned lizard that is bigger. It's about 3.5 to 6 inches in length, and they occur at higher elevations in the mountains of the Davis and Guadalupe regions in western Texas. And you can see they have the very heart-shaped head, and their horns are very short on either side of those lobes of the heart shape. And then, of course, we have the Texas horned lizard, who has those 2 prominent large horns on the back of the skull, and they range about the same size. They're a little smaller than the short tails. And they ranged originally through most of Texas, and now they're gone through a lot of the northeastern part of Texas. So current population status of Texas horned lizards. This is a map from iNaturalist, which is a citizen science program. If you've never heard of it, I recommend it highly. It's really cool to be able to go there and look at species and learn about their ranges, also kind of identify things you may be looking at out in nature. But anyway, this was downloaded in April, and it shows you the red squares are basically current observations of horned lizards throughout their range. And then in this pink zone, you can kind of see maybe this is their general their range of their populations, but you can see throughout this whole entire region here, they probably aren't found here anymore. And then you have these little nodules over here where they've been identified in Florida and South Carolina. We know that these are introduced populations, and also sometimes you get like little reportings, like here's one from Arkansas, one way over here in California. Those are probably lizards that maybe were transported there and later released or something. But for the most part, because of their wide range across several states and into Mexico, they're listed through IUCN as a species of least concern due to their size, population size. But they do have those marginal declines like we're experiencing in Texas, and it's something that we need to pay attention to and watch very closely moving forward, so that we have horned lizards on the landscape for many generations. So Parks and Wildlife started looking at these declines for horned lizards, and so back in 1998 they started the Horned Lizard Watch, and I know many of you are probably familiar with that, so I'll just kind of run through this real quickly. But from 1998 to 2006, they tried to do annual reporting for where horned lizards were seen in the state, kind of where they were found, so what types of habitats they were in, and it was, most of the reportings were coming from native grasslands or improved areas, and that's all kind of shrubby grasslands with low-lying shrubs. And then the soil types where they were found was primarily sandy, loamy soils with some clay. And then this was from the last survey work. So like between 2004 and 2006, you can see that in these green areas, horned lizards were not cited during those periods of time by any biologists, not reported by citizen scientists. And then these little brown areas, they were reported by some people, but again, these are kind of the populations that are on the margins where these animals are kind of disappearing. So when did they start disappearing and why? So again, like about 576 people responded to this question, and they started noticing these declines occurring as early as the 1960s, but a lot of them, they remember occurring between the 1970s and 1980s. In the 1970s, there were also a lot of amphibian declines that we started seeing well into the 80s, and a lot of that occurred in relation to increased insecticide and pesticide usage, or we saw those declines occurring during that period of time. And so we don't really know why the horned lizards have declined. There's never been any one reason pointed out specifically, but potential causes that have come up are, of course, habitat loss and habitat fragmentation. Those two things are probably the cause of most amphibian or most animal declines. Of course, when you start putting in roads, homes, businesses, you're putting concrete down, getting rid of the habitat, there's no place for those animals to go, and their populations become reduced to the point of not being able to sustain themselves. And of course, agricultural practices, so habitats were changed to more monocultural systems or overgrazed areas, so you don't have those as much diversity on those habitats. And then red imported fire ants introduced. They outcompete a lot of other ant species or species that the horned lizards may feed upon, also kill horned lizards. And then, of course, in the environmental contaminants, the pesticides, insecticides, and any combination of those factors. So the Fort Worth Zoo originally started working with horned lizards in 2001 when we were building our Texas Wild exhibit. So we were exhibiting a lot of native species and telling their stories, and how land managers and people can coexist with our the animals that we have in the state. And horned lizards are historically hard to maintain in captivity. We've had horned lizards in zoos as early as like the 1850s. And then like the Bronx Zoo, I think, got some like in the 1890s, but they were hard to maintain because of their high metabolisms, and also their UV light requirements. They're big baskers, and they need to be able to thermoregulate very well. And so the Fort Worth Zoo really helped establish successful husbandry methods for horned lizards through time, and we know now how to take care of these little guys in captivity, and kind of what it takes to keep them healthy. We first captive bred these guys in 2005, And then we manage the largest adult horned lizard population under human care. Currently, we generally have about 80 adults. Sometimes that goes up to 100. And so that's a lot of horned lizard mouths to feed and take care of, but we are happy to do that. Between 2002 and 2009, people were collecting horned lizards or accidentally bringing them home, not knowing what they were. And they were either confiscated or rehomed to Fort Worth because Parks and Wildlife didn't feel comfortable just putting them back in areas where they may not be able to do well, to adapt to, or may not be from the appropriate genetic population. And so in 2010, we started talking about trying to establish reintroduction methodologies for horned lizards. And historically, reintroductions for reptiles in general have been difficult and not successful. And that's because a lot of those also use translocated animals, meaning moving animals from one site, wild site, to another wild site, and then just doing basically what's called a harbor release, where you pick them up, you move them, you put them in the car, you move them to another spot, and you put them down on the ground, and then you hope that they fare well and maybe monitor them. But a lot of times these animals have really strong homing urges, and so they will just travel and travel and travel until you just can't find them anymore and likely die. So those don't always work out very well. So we also needed to determine where these horned lizards came from originally, and if populations were genetically distinct from one another. And if you can see at this map, the geological map of Texas, everybody knows Texas is a very large state, and we have lots and lots of different habitat types. And so therefore, some of these lizards could be adapted to living in very harsh desert environments, as opposed to the prairies, shortgrass prairies, as opposed to higher elevations in mountains. So we needed to look at that. And TCU is right up the street from Fort Worth Zoo. So we contacted Dr. Dean Williams, and he was happy to jump on board and has done a lot for us in this program. And he started looking at horned lizards across the state, and also some animals in Colorado and New Mexico. And he determined that there are basically three major clades of horned lizards that should probably be kept and managed differently or separately. And so the western clade, you can see is kind of this area here within the Chihuahua Desert. And then the northern clade is everything up in here. And then the southern clade occurs further down the southern part of Texas, below the Balcones escarpment. So currently, right now, what we work with is everything that comes from this region in the north central part of Texas. And we also genotyped the captive animals we had at the zoo. And there were a couple that we had from those previous donations or confiscations that came from the western clade and also the southern clade. And so we gave those to other zoos to work with. There were only a handful of animals. It wasn't that many. So it really didn't affect us much after we did the genetics work. So that was a good thing. And in 2011, we formed the Texas Horned Lizard Conservation Coalition, and we meet twice a year. It was a forum for biologists and local agencies that are working with horned lizards in North Texas and Oklahoma to kind of get together and share current information and talk about our different projects and identify conservation priorities for the species as a whole, and then also kind of explore collaborative resource allocation and options that we may have, and also more importantly to detect gaps in research so that what we're doing is successful and we're not overlapping a bunch of work. So for that first meeting we had Chip Ruthven from Texas Parks and Wildlife come. He's a manager out at the Matador Wildlife Management Area, and he's been doing a lot of work for decades, mark and recapture study on wild horned lizards there at the site. And he's captured well over, this is kind of old information, but well over probably close to 4,000 animals by now, I'm sure. And he helped us understand kind of land usage, ages of animals, and also kind of where he was finding them, what kind of habitats they were occupying. So again, you can see they kind of like those loamy sandy soils. And then we also invited Ray Moody from Tinker Air Force Base. He's a resource biologist that's hired there. He's been working there for many years, and he's been looking at a population of horned lizards that occurred at the base and had to be translocated or moved because of a housing development on the base, and then also was looking at what the impacts of construction have on these horned lizard population, on that horned lizard population that occurs at Tinker. And so Ray's always brought some great information to us from his project. We've learned a lot from his project, and also especially about new technologies and how to track these little tiny horned lizards, hatchlings, because before the radio telemetry equipment that we have, the transmitters are larger and heavier, and so we can't put those on the little guys. And so he's been very helpful with us with our project, and we're very grateful for that. So in 2011, Fort Worth Zoo started a pilot study in a private land in Parker County. It was a small property, and our primary goal was to establish methodologies for introduction of horned lizards that were hatched at the Fort Worth Zoo. And it was a 60-acre ranch, but it was surrounded by other properties that were much, much larger. So if we were successful, we knew our population could expand into those other areas. And they had a high harvest rate density, and they were also going through a lot of restoration work, bringing in new native plants and grasses. And they were managing the site organically, so not using any pesticides or insecticides. And they had about 10 to 15 head of cattle, so they were rotating those through the pastures and again helping with that native grass restoration. And the last horned lizards that were actually observed there were 30 years ago, and it was within about 70-mile radius of the zoo, so we could get there and drive back and forth and monitor these guys on a daily basis. So we put standard radio telemetry tags on our adults. We first started with just a pair, and they did really well. They survived through the first winter. We learned a lot about hibernation, also learned about home ranges. When we used the soft release method, we put the adults in those cages, and it allowed their natural prey items to go back and forth through the cage, but then it also allowed them to orient themselves to the property. And that way they didn't run off like you would normally see with lizards that are normally just hard released. And then 2 years later, the Parks and Wildlife wanted to duplicate what we were doing, but look at the feasibility of using translocated wild-caught lizards instead of captive lizards. So basically using the same techniques we were using, but then using the translocated animals to see if the methods would still work with those. And so they were, again, collecting wild horned lizards and then moving them to the Muse Wildlife Management Area, just northeast of Brownwood. And they came out to our site, and I just want to give a heads up to this gentleman, Devin Erkleben, and this one, Nathan Rains, who was supposed to help me with this talk, but I don't know, he's off doing something fun, I guess. Not that this isn't fun, but you know what I mean. But anyway, these two guys have really been instrumental in everything that we've been doing, and it's been really great working with them, and we continue to work with them, so I wanted to make sure that they were mentioned in this project or this presentation. During this time when we're moving a lot of horned lizards around, we decided we needed to really look at the health status of both wild and the captive horned lizards, because nothing's really known about wild populations of horned lizards in their health. So in 2014, a veterinary student, Michael McIntyre, did his internship here, and that was his project. So we helped screen wild and captive horned lizards to assess their overall health and prevalent parasites and diseases. And we wanted to understand any potential disease risks that would affect any reintroduction programs using both wild and the captives. And then, of course, we were going to use any of that information to help inform other partners or other people in terms of thinking about these things when you're moving animals from one place to another. So we did full exams on all the animals that were translocated to the Muse Wildlife Management Area, also at the zoo, which included x-rays, and then full workups, blood, feces, and then also some viral screening. And what we found was, and what we were able to do out of this project was actually develop baseline parameters for blood values for wild and captive horned lizards. And then also started looking at parasite prevalence of wild lizards. And you could tell here in the red bars indicate individual fecals, and the green bars are group fecals. So we took animals, groups of animals, and ran them because of limitations of funding. And so the green bars really are what we're looking at here is coccidia, mites, and the nematode parasite for the horned lizards were very common and not surprising to find. The coccidia is found in a lot of invertebrates and transferred to the animals that are eating them, such as crickets. We find that a lot in captive situations. And then also external mites. We would find mites on horned lizards when it's very dry and we're going through droughts for some reason. Seems when we find them the most. And then also that parasite that lives in the horned lizard's stomach and then is actually defecated out through its feces, eaten by a harvester ant, and picked up and put in and fed to their larvae, and then it pupates in and infects the harvester ant, and then again is eaten by the horned lizard later. So a really interesting relationship between those, but not surprising and not something to worry about in horned lizards really. And then salmonella was detected in 7 of the 49 of our samples that we ran initially. Again, not surprising. Reptiles carry salmonella, but it's not usually anything to be concerned about unless they're immunocompromised. And then 15 different bacterial groups were isolated, and I know some of those were newly described in horned lizards, but nothing to be concerned about. And then we did not find any of the typical viruses that we may be concerned with reptiles, so the adenovirus or the herpes virus. And in 2015 to 16, Mason Mountain started to mirror what they were doing out at the Muse. Again, Parks and Wildlife collecting horned lizards, wild horned lizards, and then moving them out to the Muse to see if they would fare better out at the Muse, or I'm sorry, out at Mason compared to the Muse, because Mason had more naturalized habitat than the Muse did. And so we wanted to see if there's any differences there. So outcomes from all those projects between 2011 and 2016, we learned a lot about the spatial use and survivorship of translocated lizards, and as well as lizards that are released from captivity. And we know that just about anything will eat a horned lizard, of course. Bobcats, skunks, raccoons, hawks, kestrels, shrikes, roadrunners, and especially snakes. Copperheads, we documented a copperhead that ate one of our horned lizards out at the private ranch with the transmitter. And I know the guys out at the Muse did the same thing. Found transmitters in coach whips for sure. And then also we realized predator control is definitely necessary when you're trying to do these things. We would find horned lizards with their heads chewed off, and we think that that's probably likely rodent predation. And we also found out that a single fire ant sting can kill a hatchling horned lizard. Until they get to be about 5 or 7 grams, they really can't fend off that venom that gets injected into their system. And they also like to congregate in groups. We found at our private site, and so if they're all huddled together and a single fire ant comes in, this is what we would find within 24 hours. This happened. We had 13 horned lizard hatchlings just demolished, which was unfortunate. We found that captive hatched and wild translocated lizards both adapted very well using the soft release methods. We didn't have any problems once we opened those cages with horned lizards running off and never coming back. And then we learned that horned lizards in the wild and in captivity are generally healthy, and that there is nothing that was identifiable that we should be concerned about in terms of when we're moving things around. And we know survivorship is a numbers game. We lost between 75% and 90% of our animals from both the wild translocated and the reintroduced populations, which isn't surprising, but very sad. So with all that information we've learned, we've kind of dialed it back. We know that we've got to focus all of our resources to one project because we've got to be able to put out the numbers to kind of stay ahead of the predation game. And so we started focusing on the Mason Mountain Wildlife Management Area for our projects, and that's what we're currently doing. So I want to kind of back up a little bit and talk about our Texas Native Reptile Center at Fort Worth and kind of how we manage the horned lizards here and get everything ready to go for reintroduction. We typically house our lizards most of the year in these outdoor runs. And it's important because, again, these guys like intensive ultraviolet light, so they do bask. And they also need proper thermoregulation. So they need to be able to get out of extreme heat and thermoregulate on their own. They have very high metabolisms, as I mentioned previously. They eat a lot of food, so they've got to be able to warm their bodies and move that stuff out and keep their digestive tract moving. So we keep them these 4x8 wooden enclosures, and this is an older photograph. We do divide them now into 4 different compartments because we have so many lizards. And the sand substrate's about 10 to 12 inches. And we plant them with live plants, rocks, and cork bark. And then we have a misting system on the enclosures, and we use shade cloth as well because horned lizards, believe it or not, even though they can be out there in 100-degree heat, they do overheat. So that's important. As mentioned previously, we house usually about 60 to 80 adults, sometimes up to 100. And we identify all of our horned lizards as individuals. We take photos of their bellies. They're all unique, and they stay that way. And then we also, in the past, have used fingernail polish to give each lizard a unique color combination on its back so staff can instantly view a lizard and know that who it is without having to pick them up and disturb them. And each lizard has its own house number, that 206986. It's marked with the mail. And then we track everything. They each have their own medical records. They have their records in terms of their weights, their behaviors, breeding. Staff watches these guys at least 3 times a day, goes out and feeds them 3 times a day. So we collect a lot of information on them. In the diet, our adults primarily feed on red harvester ants, but we also supplement their diets with crickets, dermestid beetle larvae, and waxworms. And then the hatchlings, we start off on termites because we found that that's what they do best on, and they'll start to grow a lot more quickly on termites. And then they will start to eat on things like fruit flies, pinhead crickets, bean beetles, and harvester ants as they get big enough to be able to fit those in their mouths. And we put the harvester ants in these little deli cups, and then are partially buried in the sand so that the lizards, like the one on the left there, can just go right up to the stick and they know where dinner is and they can just peck those off without getting stung. And then sometimes when we need to bring lizards indoors and we want to make sure they're all getting their fair share of meals, we will hand feed basically the lizards. So we'll take some of the harvester ants and on a spoon and drop them in front of the lizards, and they sit there and we'll nibble them off. So they don't really have to hunt a lot for their food. life of luxury. So they eat a lot. Hatchlings and juveniles typically eat 20 to 50 items per day. And our adults eat usually 50 to 100 to 150 items per day. And you can see in this photo, this is an adult lizard, and you can see the size of the feces coming out of this lizard. I mean, you can tell they sit and eat a lot of food. And then if you break that open, you'll see a lot of exoskeletons. It's very dry. It's not moist at all. It's just very dry, and it's just full of exoskeletons. And this is a photo of a feces from the wild, and you can see mostly primarily harvester ants, and there's some beetle wings in there. Through October through March, our horned lizards are hibernated. We allow the horned lizards to kind of show us when it's time, so they're outside until it gets cold at night. They'll start to go off feed, and then they'll start to kind of cover themselves, so we know that they're shutting down. And we get we used to use these modified freezers, so standard upright freezers that we put new thermostats in. And then we kind of ran out of space with those, so we graduated to a big walk-in cooler, which we use currently. Horned lizards, we make sure that they're fasted 7 to 10 days prior to hibernation. There are a few that don't go off feed before we bring them inside, so we'll bring them indoors, and then we'll make sure they're fasted. If we don't fast them, they will die from bacterial infections. And then we cool those down gradually, usually about 5 degrees per day through a 7-day period. And we maintain them between 45 and 50 degrees Fahrenheit during the winter time. And then the horned lizards, again, signal to us when it's time to wake up. They will start to come to the surface, move around. They're not moving around very quickly, but we can tell that they move from one spot to another because staff check them every day. And then it's really curious because when we first started our project out at the private ranch, we had animals that we were monitoring through hibernation, and then we had the captive born animals inside. Those animals would all wake up at the same time, despite being outside or inside the cooler. So they have this very strong internal clock that tells them when it's time to go down and when it's time to come up. So that's very interesting. Breeding occurs April through May. Once we bring the animals out of hibernation, we make sure everybody's healthy. They have physical exams. And then we make sure everybody gets to an appropriate weight and are moving around, eating well. And then we'll put the males, typically one male with one female, but sometimes we'll do trios, one male, two females, depending upon our space limitations. And then we just kind of watch them, and we try to observe first date of copulation. The males will just line up with the females. If the female's receptive, she'll stay on top. She'll allow the male to mount her. If she's not, she'll hunker down, bury herself, or she'll turn around and bare her teeth, I guess. And they do have teeth. Bare her teeth a little bit to him and say, take a hike. And then, but they do use the horns and hang on to those. And so when we're doing pairings, we look at genetically appropriate pairings, but we also have to look at the size of the animals, the weight of the animals. So we try to pair them up as closely as we possibly can. So if we have a really, they might be close in weight, but if the male's not as long as the female, he might have a very hard time breeding with her, and so we'll end up with infertile eggs if we try to force that pairing. So we're very we watch that a lot when we're pairing everybody up. Egg laying occurs in May to June. Typically, females will dig to the bottom of the enclosure to lay their eggs. They like to pick out the moist areas, kind of alongside the walls, alongside the potted plants that are in their enclosures. And then they typically lay about 1 to 26 eggs for us. The average is 13. We have had a maximum, in the stub book, the maximum number of eggs laid was 50 at one point. And it's all pretty relative to the size of the female. Those bigger females, you will get larger egg clutches. But for those The smaller ones, you know, average is 13 for us. And then they usually lay about 1 clutch per year, but some of those bigger females or females that have bred first thing in May will double clutch. So they will lay a second clutch of eggs towards they usually hatch in September, October. This is a video of a female digging. They kind of go in head first and start digging a hole till they get to the bottom or where they're comfortable, where that temperature or that moisture content is appropriate, and then they'll turn around and lay their eggs and move out. And they're very good at covering the site to where you can't even tell they dug there. Staff, again, check them very regularly, so they will see females start to do test hole digging and get a sense of that the female's going to dig. Usually they dig late afternoon, and we will find eggs have been laid by the next morning. We don't always see the females digging, so staff, we weigh our females regularly, weekly, and if we see a drop in weight, then we know they've laid eggs, and we dig them up. And we dig them up because early on we used to leave the clutches in the sand, but they would desiccate and dry out very quickly, so we do incubate the eggs. This is one of our keeper staff digging up the eggs. They do that very carefully till you see them, and then we move them to a container. So females do lose about 30 to 40% of their weight after egg laying. So they have a lot of eggs. And eggs are typically laid 2 to 3 weeks after breeding. And then, as I mentioned, we put them in, we're careful we move them, don't flip them over, and we put them in dishes, deli cups with vermiculite. And we also use what's called Sims containers, where that's a photo of this, where the eggs are like suspended over water. We put substrate in the bottom of our Sims containers because we don't want juveniles squeezing through and drowning. We weigh all of the eggs, we keep data sheets on those, and then we incubate them between 83 and 84 degrees Fahrenheit. That's the temperature that we have found to be the best through the years. And they are not temperature sex dependent like some species. So everybody comes out usually about an even sex ratio, male to female. And hatching, again, occurs usually July to September. Most of them will emerge in July. And we do have some that, if they double clutch, we'll get those late September, early October hatchings. So they're very tiny when they hatch. The eggs are about the size of it, almost like a fat tic tac. And the eggs hatch within 6 to 8 weeks after being laid. And the hatchings weigh less than a gram. And they fit pretty much on the building on the back of a penny. And the entire clutch will hatch within 24 hours. So typically once the first egg hatches, I think the other siblings can sense that. It probably walks around on the eggs, and they all hatch out, and then they'll all emerge, if they're buried, all emerge at the same time. Hatchlings are really prone to dehydration, so we mist them twice per day, and we also soak them in water shortly after hatching. And then we also put like wet paper towels and moss within their enclosures to make sure they always have these moist micro climates to go into. This spring, we were able to renovate our greenhouse. So we are expanding our capacity to be able to breed and maintain horned lizards. And hopefully we will try, I think, hibernating these in the greenhouse this year. So we may not use the coolers. This will also give us more capacity to breed, I think, early on in the spring. Because when we put them in those outdoor runs, you're kind of at the will of nature. And sometimes we get those cold springs and our animals can't get outside as early as we like. So hopefully that will help with that. And our current reintroduction focus is out at the Mesa Mountain Wildlife Management Area. We release 14 to 30-day-old hatchlings. To date, we've released a total of 989, so close to 1,000 hatchlings out there. 712 of those came to from Fort Worth. Dallas Zoo joined, and we have they've given us 242 eggs. And Caldwell Zoo recently joined the program and have started reproducing animals and produced 35 hatchlings for us to date. So what are our challenges with this? Well, the more partners that join is the more competition for harvester ant food, and for us to be able to rear additional offspring for release. So that's kind of a catch-22 for us. There's only one supplier of harvester ants in the country. And a couple years ago, he was pretty much maxed out. And so we really can't have a lot more join unless they can find their own harvester ants, which can be difficult and very time consuming. The other thing that I've mentioned over and over again, predation will always be there. And it's something that we need to consider. It's a numbers game for reintroduction, and red imported fire ants are also an issue. And then, of course, monitoring. We need to do monitoring. We need funding to do monitoring so that we know what we're doing is successful or not, and how we can keep adapting for success. So kind of what have we learned so far? In 2020 and 2021, we brought a PhD student, graduate student, Rachel Alanis, onto the project, and she had already done some horned lizard work with Dr. Dean Williams down looking at some horned lizard populations in Kennedy. So she's very familiar with the critters and did a really great job. But we're looking at the influence of location and release method on short-term survivorship of hatchlings that are reintroduced into Mason. That was our first project. So Rachel took belly photos of all of the hatchlings, again, for identification purposes later. And then we outfit all of our hatchlings with these harmonic radar tags that I mentioned. So the way these work, they're passive, so there's no battery involved. That's why they're so lightweight. They originally created for to find avalanche victims. And what it is these tags can be sewn into the skier's clothing, and then the emitter, which is the box on the left, emits a signal, a radar wave, which is then reflected back from the diode, which is that little centerpiece right here, and then you get the beep. So it's very much like a bat submitting a frequency that bounces off of an insect that it's tracking. It's a passive way of finding something. And the diode tag, although it's very long, it only weighs less than 0.05 grams. And the reason, and we've tried to cut the length on these, but this is the antenna on both sides. And right now, as the tag is, it will only pick up a hatchling from about a 10 to 15 foot distance. Okay, so it has a very short range to begin with. And if we cut it any further, I've tried, usually you can only pick these guys up within 5 feet. So it renders the technology kind of useless if you cut that antenna. And I know our partners out at Tinker have tried using some different methods to shorten the antenna, and still have had issues even when they put them in like a spiral. They catch on the grass and stuff. So we found that this is the best method thus far in order for us to track these guys and learn how to be successful. And we spray the tags so that they blend into the environment, and then each tag is labeled with an A1, or like A0 through A9, B0 through B9, so that again, you don't have to pick up the lizards when you're out there tracking. You can see the letter and know who you're following. So we wanted to see what the best release method for hatchlings were. We're doing basically a hard release with these guys, but we're comparing whether or not we release as clumps or dispersed. And we originally started releasing in clumps because that's how they come out of the ground when they hatch, and that seemed logical. But then we started to see some weird things at this site where we were releasing clumped animals. So we're like, well, let's do a study and figure out what the best method is or what the differences are. So in 2020 at site 1, we put animals in a dispersed pattern, again using this grid, and about 5 meters apart
and there's 100 points, and then compared that to site 2, we did a clumped at site 1 the following year. So we flip-flopped it. And what we found, and then Rachel went out and would track these guys, of course, up to hibernation and find out what happened. And despite the close proximity and the similar habitat, these sites are very similar in habitat, again, because they're so close, 300 feet, within 300 feet of each other. The hatchlings that were released in groups or clumped seemed to disappear very quickly. And I mean, they would disappear overnight. So, you know, we couldn't figure out what was happening. Most likely that might be a predation events, but we don't know that for sure. And that hatchlings released as singles or in the dispersed method survived longer, and they survived longer at site 1 than at site 2. Okay. Overall, the survival rate through the first winter of hibernation, or up to the first winter, was 26%, which was comparable to the 28% survival that they're seeing with wild horned lizards that reproduce out at the Tinker Air Force Base. So we're very that's very optimistic for us. So promising milestones that we've reached thus far. We have survivorship of hatchlings to adults. We have documented reproduction every year at the site. We have egg laying, and we have F1 hatchlings that have been produced out at this site. So exciting news for us. And we theorize that survivorship is correlated to the release method, so that clumped versus dispersed, and or the presence of red imported fire ants and their effects to the horned lizards or the hatchlings' food sources. So site 1 actually doesn't have very many fire ants, but site 2 does. So how do we reduce the impacts of red imported fire ants, and how do we determine what's occurring out at both of our sites? So we decided to investigate the utility of using targeted pesticide treatments for fire ants, and we do that with Amdro. And we expect our immediate results to increase hatchling growth and survivorship. And then long-term benefits, we hope, will include the completion of a targeted fire ant treatment plan for horned lizard conservation programming moving forward. We'll never get rid of hot fire ants entirely, but how can we manage for them? So we set up 4 different sites for this study. There are 4 50 by 50 meter sites. We have our original release site, site 1 and site 2. Again, site 2 has more fire ants than this site does. We have site 4 is a control, and then site 3 as a control. So each site's a 10 by 10 grid of flags, 100 meters apart. So there's 100 flags at each site as well. So 400 sites, or 400 flags total. And we utilize a 2x2 study design, as I mentioned, 2 treatment sites where the Amdro is applied, and then 2 controls where no Amdro is applied. So we have our control here. So no Amdro is applied, no Amdro is applied. We apply Amdro here and here. So we do 6 baiting surveys per site, and we did those from August 2021 to August 22 so it was spread out during the course of a year. And we put a hot dog at each flag. And then after 30 minutes, we come back and we look at the hot dogs and we identify if there's red fire ants present, harvester ants, or other. If there's fire ants present, there's not going to be any other species. And we go back after 30 minutes and we apply the Amdro to the lids where the fire ants, only where the fire ants occur. And then we go back and we cap all of the lids to identify all of the ant species later. 2 weeks post the baiting surveys, the biologists come back and they do pitfall trap surveys for the ants to determine what the overall effects are to the other ant populations, or to all ant populations, including fire ants at those sites. So this is just preliminary results, again, for Rachel's work.
and we don't have the final results. And I know that it's kind of hard to look at all this, but basically it shows that the red imported fire ants, even after baiting, you know, on our controls here and here are present. And then they're really not very abundant here at this site. And they are still abundant at site 2, but not as predominant as they were. You can't see that because you don't have the chart. But the other ants, we have the other ant category because there's some that we're really not that concerned about. Some are like a tree species, but these other ants, the cheese ants, the pyramid ants, the harvesters, the big-headed ants, and the acrobat ants are all ants that the horned lizards have been documented to eat, and we know that they're important food sources for the juveniles. So those are the ones that we're primarily watching and concerned about. So what did we learn? We learned that the populations declined over the treatment periods. The timing of surveys is important. Typically, we go out there, we get about 10 people to do each one of these surveys, and we try to get out there first thing in the morning. Well, it's on survey number 5. You can see that this means no ants were found. Well, that was because the surveys occurred much later than normal, and so that kind of skewed that data. So it's important to survey at the right temperatures. Obviously, the ants are less active during really hot periods of time or when it's really cold. And then proper identification of ants is extremely important. The big-headed ants and the acrobat ants look a lot like the red fire ants, just from the naked eye and somebody that's not a skilled ant identifier. So in the future, we're going to continue doing all the surveys in the pitfall trapping, and then we're developing an easy identification guide for surveyors or managers to use when doing this. And then we need to determine how long the treatments will be effective, and then also try to determine if this technique is repeatable at other sites and for other species beyond horned lizards. So it could be applied like with Atwater prairies, chickens, bobwhite quail, things like that. So ultimately, our next steps, we have a new PhD student that's joined the project, started already. She's looking at female nest selection and hatchling survivorship. And then we're contracting a biologist that's starting next month to assist with radiotelemetry tracking and identification of habitat preferences, and also helping with that, determine what the survivorship is. And then also comparing a wild population from a nearby private ranch to our reintroduced population. We already started kind of doing that with some students from TCO last year, looking at the different types of grasses and the soil types, and now we're going to keep doing that. And then also, of course, continue with the surveys for the fire ants. And then we at the zoo are also working on some new assisted reproductive technologies, development for artificial inseminations, semen collection, and then of course biobanking. And then we've partnered with some students at Oklahoma State University to start looking at the microbiomes of the horned lizards used for reintroduction compared to their wild counterparts. And they're also really specifically interested in looking at microbiomes, differences between what's passed from the mother to the hatchling, potentially, if there's any correlation or something that we need to be worried about that. And then in the future, of course, we need to really start looking at our primary predators. We know what predates on horned lizards, but we need to know what we need to try to control at different different seasons, and so that we can manage. Again, we're never going to get rid of all these predators or things that kill horned lizards, but it's a numbers game. If we can get the scales to balance and then ultimately kind of tip towards horned lizards, we'll be able to establish a population. And you know, it's like that with any reintroduction program. So once we figure all of this out, then we can hopefully continue to fundraise to support this project, then also explore additional release sites if those resources allow us to do that. So I just want to thank everybody, and if there's any questions, I'm happy to answer them.